Yeah, I think we are really seeing an important expansion of gene therapy trials, in particular concerning hematopoietic stem cell and T cell, actually. In hematopoietic stem cell, which is what we have been mostly developing with for the last 15 years, this is aiming at treating several genetic diseases of the blood and the immune system. I would say a key development has been a platform based on lentiviral vector, which are more efficient and also safer than earlier generation vector. So patients are showing a very high level of genetic engineering of the hematopoiesis associated with safe outcome, so no indication of abnormal clonal expansion, and very significant benefit. We have 3 trials ongoing in our institute using lentiviral vector gene therapy. 2 of them are very advanced, and they are one in Wiskott-Aldrich syndrome, which is a combined immunodeficiency and platelet deficiency, and the other is in metachromatic leukodystrophy, which is a storage disease. In both cases, patients are treated with their own stem cells, which are harvested, modified with a vector, reinfused after conditioning. And as I said, we see up to 80% of the hematopoiesis post-treatment genetically modified. And this is as long as 6 years now for the first treated patients. We have 8 patients treated in one trial and 20 in the other already. All of them are showing very high level of marking with no sign, as I said, of altered proliferation of cells, and importantly, very significant benefit. In the case of Wiskott-Aldrich, essentially, as you know, this disease can be treated by allogeneic transplant. So the patient we were enrolling in the trial were the one for which there was no matching donor available. And the patient essentially have a clinical benefit as after a successful transplant. So they have been essentially very well recovered from all the signs of the disease. And they did not essentially experience the risk of an allogeneic transplant because they were using their own cell. In the case of metachromatic leukodystrophy, this is more interesting because it's a neurodegenerative disease. So why would you treat with a hematopoietic stem cell therapy? Because there is a rationale of some of the stem cell to home in the brain during the transplant procedure to engraft as microglia progenitors, and they can repopulate in part the local population, microglia and macrophages. And this can help to clear the storage. That's why many storage diseases like mucopolysaccharidosis, Harler, are also undergoing normally stem cell transplant for treatment. But in case of leukodystrophy, this is very, unfortunately, unsatisfactory, so it's not routinely employed. We did in the mice a test in which because of gene therapy, we can overexpress the therapeutic enzyme in the stem cell. So when the stem cell home in the brain, they also release some more enzyme, which may cross-correct the other cell in the brain. And what we have seen is that as long as we treated patients very early in the disease, we have prevented disease development today. So essentially, the patients are doing very well, while they would probably be, unfortunately, dead now because of the disease. So it's quite a significant benefit that stem cell therapy is giving. So that's, I think, is making a case that there are, in principle, important strategies, both where stem cell transplant is working, but then you could not have a properly matching donor, and then you can use own stem cell for the patient, or in case where there's no obvious treatment with normal stem cell, and by gene therapy we can make this cell for instance, increasing gene dosage, and then become effective. While this gene therapy is being tested in the clinic, what we are doing in the lab is now using gene editing approach, which is the new twist in the genetic engineering approach. Gene therapy, as I discussed in the clinic, is using vectors which still semi-randomly integrate in the genome. There's always some risk where this vector goes to integrate. Even we are not seeing, as I said, adverse event today. Editing instead means very precise genetic changes. So we essentially direct either the insertion to a specific locus, which is safe, or we rewrite the gene to correct the mutation. That's why we call editing. So we essentially rewrite the letters there. And this is using artificial enzyme, the most famous one being CRISPR now, but we have been working with zinc finger nucleases and talents before. And this is a way by which we can actually make a true precision medicine. It's early days. It's not yet in the clinic with haematopoietic stem cell. It is in the clinic with T cell now, so we'll probably be soon seeing the first clinical testing. It may be the new approach.